Welcome to the lecture of part 1 of non-Mendelian genetics, focusing specifically on incomplete dominance and codominance. And so the crosses that we've done in the past are crosses that Mendel found, and so that's why we call them Mendelian genetics. And now we're going to look at a couple of different crosses that don't follow Mendel's rules, that happen to have some different traits. And so the first one we're going to look at is incomplete dominance. Incomplete dominance is when one allele is not completely dominant over another. And since they aren't, they end up blending. And so here we have a homozygous dominant flower that's red, so they would have 2 capital letters here. We have a homozygous recessive flower over here that would have 2 lowercase letters, which makes it white. And then when we have our heterozygous flower, our hybrid here that has a capital and lowercase, instead of it being red, it's now going to be pink, because in incomplete dominance, those 2 colors are now going to mix together to create pink. And we see this a lot in flowers. And so now we're going to look at carnations to practice an incomplete dominance problem. So in carnations, the color red is known as incompletely dominant over white. The hybrid color is pink, which is RW. And so if you notice here, we're actually not using capital and lowercase letters because both of them are seen as dominant colors. But we do see that we use 2 different letters, R and W, making the red color capital R, the white color capital W, and then the pink color is going to be capital R, capital W. Then I'm asking you to give the genotypic and phenotypic ratio from a cross between 2 pink flowers. So we're always going to start with our alleles. Our alleles are going to be what's up here. R is for red, W is for white, and RW is for pink. Next in our step is P for parents. Now we need to find the parents. And the parents are 2 pink flowers. The only way to create a pink flower is for them to have this RW genotype. And so you're going to end up with an RW over an RW here. Again, our parents are RW times RW. Place them on the top and the side. Go ahead and fill out my Punnett square. So now I have the possible offspring inside my square. Now we're going to first look at the genotypic ratio. You remember genotypic ratio always is talking about the letters. And so you're going to have 1 RR, 2 RWs to 1 WW. And then the phenotypic ratio is talking about the physical characteristics. So you have 1 red to 2 pink to 1 white. So that was an example of incomplete dominance. Once again, incomplete dominance is where they're physically mixing together. So the heterozygote is always going to be a mix of the 2. Now the next one we're going to look at is codominance. Codominance, once again, you don't have a recessive gene. Both of them are dominant. And when both alleles are present, then both alleles are expressed. So the homozygous pink here would be 2 capital B's. Then the homozygous white here would be 2 capital W's. And then if I have a capital P, capital W, I'm going to end up with this flower that has white and pink expressed because both of them are being expressed as the heterozygote. Same for this roan cow. A red cow would be 2 capital R's. A white cow would be 2 capital W's. But this roan cow, which is a cow with the red spots and white splotches, is going to be an RW. So let's practice one of those. So in certain species of chickens, black feathers are codominant with white feathers. Heterozygous chickens have black and white speckled feathers. Cross a black chicken with a white chicken. And so first our step A, alleles, we already have them here. Capital B is black, capital W is white, and then the BW is speckled. Next, I need to find the parents. I'm crossing a black chicken, which the first parent is going to be black, capital B, with a white chicken. It's going to be 2 capital W's. I'm going to place it in my Punnett square for the next P, fill in that Punnett square, and now I'm going to find the genotypic and phenotypic ratios of each of these. So, I'm going to do the genotypic ratio first, which is the letters. You'll see that I have 0, capital V, I have 4, capital B, capital W, and 0, capital W, capital W. Then for my phenotypic ratio, you can see that I have 0 black, I have 4 speckled here, and 0 white. So once again, when you have one of each codominant pair, you're going to end up seeing both of them present. The difference between incomplete dominance and codominance is incomplete dominance. They're blending together. So it's like blending of colors. You end up with a mixture of the two. And then the codominance, both of them are going to be expressed. So you're not seeing the pink mixing with the right or the red mixing with the white. You see both of them physically present. So from here, you're going to practice some incomplete and codominant problems in your practice packet. You're going to do that for homework tonight, and they're going to be due before class tomorrow.